Okay, so welcome again. So thanks for joining us tonight. We are going to jump. We've been doing introductions. Our next meeting is June 14th with topic is TBD. We're going to have a mixer next Thursday at Wing Walker Brewery in Monrovia from 6 to 8. Informal meeting, no presentation, just getting together with the local tech community. If you have any feedback on this program or other program or ideas for other programs, email us at email@mediatech.org. With that, I will go ahead and introduce our presenter. Just a quick word about Medea. We're a 501c3 nonprofit organization, and we do educational programs such as this in order to unite the tech, industrial, and scientific community here in this area of San Gabriel Valley. And this one is on entrepreneurship, and we also have them on business finance and development and operations. So that's what Medea does. Our presenter is Dr. Kevin Scanlon, who's had a career spanning roles in academic medicine, senior management in the pharmaceutical industry and biotech communities or companies, an author as well as an individual investor. His book, The Hybrid Entrepreneur, addresses key business issues faced by new ventures and is available on Amazon. He served as the chairman of Pasadena Angel Investment Group in Pasadena. He has been CEO of 4 biotechnology companies, including Melanoma Diagnostics, which was sold to Myriad Genetics. And he was at Schering AG in Germany before that, or after that. He was responsible for the genomic and cancer programs in the US, Europe, and Japan. Dr. Scanlon's notable academic achievements include a Leukemia Society Scholar, awarded the Paul Martini Medical Research Prize in Germany, co-founder of the Nature Journal Cancer Gene Therapy, co-authored 139 peer-reviewed scientific articles and 9 medical books. He has 7 issued patents. So please welcome Dr. Scanlon.
Thanks, Bruce. Let's see. I'm going to share the screen, hopefully. I've broken up the talk into just 9 slides, so it shouldn't take very long to go through my background in science, the pharmaceutical industry, and investors, and where I think the biotech industry is going in the next 10 or 20 years. And then we can take questions after that. Okay. So I was born in New York City, but I have an Irish passport. That's a long story. I was fortunate enough to go to England and get trained in molecular biology. And I came back to the United States, and eventually I got trained in business in Berlin, Germany. I've lived in Pasadena for about 20 years, and I moved to Boston about 8 years ago and just returned to Pasadena. I think it was through divine intervention that I wound back up here in Pasadena. So my background in science, I was planning to be an English professor in London for the rest of my life, and I would be retired now. But the chairman of the department at the University of London said that I was pretty good at doing research, and the money really was in the United States. And so if I really wanted to do research, it would be better for me to go back to the United States, although they had a position for me in London. So my research had been on how messenger RNA is regulated. And at that time, everybody wanted to study DNA. So I was in a course at Harvard. There were 120 students in the course. 119 wanted to do DNA research. I was the only one who wanted to do RNA research. So you can see I've always gone against the grain a little bit. I finished my degree in England, and I went to Yale, and I learned cancer pharmacology, how do drugs work, how do patients become resistant to drugs. And I had a really good boss advisor who was an authority on cancer and drug resistance. And he wanted to learn what I was doing in molecular biology, and I wanted to apply my molecular biology knowledge. After Yale, I went down to Mount Sinai to work with Jim Holland, and Jim Holland wrote the book Cancer Medicine. It's about 2,000 pages. So I felt that he was the best person to learn why drugs don't work in patients. And I went back to trying to understand why cancer chemotherapy agents don't work. And I was very fortunate because we were able to identify a couple of novel insights into how cancer cells work. And I proceeded to publish 4 PNAS papers in 7 years. So that was the good news. The bad news is I didn't get my NIH grants renewed.
Don't forget to take out your trash cans.
That, I always forget to take out the trash cans. So I was pretty lucky, and I wound up getting a research prize in Germany. And ironically, the committee, it was my first exposure to the German culture and business community. They, one of the people there was a doctor in Kiel, Germany, that was very interested in the work that I was doing. And he'll come around when I work in the pharmaceutical industry later. But I won the award. So you've all familiar with the RNA PCR assay for COVID? Yeah. I published the first paper on RNA PCR to detect drug resistance in patients. And I got the first patent on it, although I didn't make much money on it. But so that was my interest. I was doing RNA work. I was looking at how cancer cells became resistant to chemotherapy agents. And we developed some new gene therapy type technologies to overcome drug resistance in cancer. And in the process, I co-founded a journal called Cancer Gene Therapy. And I then became president of the International Society of Cell and Gene Therapy. So that's kind of my background. It's molecular biology, it's cancer. Why do drugs work? Why don't they work? So the person that was funding my research was at Bristol Myers Squibb Pharmaceutical Company, and he moved over to Schering AG. And the company was out of Berlin, and the American component of it was called Berlex. And Dale Stringfellow had known me for about 10 years, had liked the work that I had been doing, and asked me to run the cancer program. So I told him, look, I'm a scientist. I have my own building. I have like 10 or 15 postdocs working for me. I don't know anything about the pharmaceutical industry, much less running a whole. He said, don't worry about it. We'll mentor you and make sure you don't fall down too many times. So that was my first break into business. And it opened my eyes to how much I did not know and how much the pharmaceutical industry was involved in taking a concept to a product in the market. And so that really opened my eyes. So the program was involved in cancer research and genomics. And I was supposed to supervise the group in Berlin, San Francisco, and Osaka, Japan. I did that for about 5 years. We were pretty successful. We used the technology I had developed in academics to study human cancer patients, and we identified new cancer genes and patented them. And we developed some new cancer diagnostic assays. So the amount of money that they put at research was just unbelievable. So that was a true learning experience about what they can and cannot do. So after 5 years, I came back to Pasadena. I'm always coming back to Pasadena. I don't know why. It must be something in Pasadena. So I came back and I met Mark Lieberman at breakfast one morning through a mutual friend. He was running the Business Technology Center, explained to him what I was doing. And he said, you've got to meet Dwayne Peterson and Al Schneider. So of course, a couple of weeks later at Green Street for breakfast, I meet Dwayne Peterson and Al Schneider. And they say, you can speak science and you can speak business. You're joining the Pasadena Angels. So Dwayne and Al were really, really good mentors. John Morris and Richard Morgenstern heard about me and said, no, 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 no. You can't just join the Pasadena Angels. You have to join the Tech Coast Angels. So I joined the Tech Coast Angels with Al. Al Schneider and I went back and forth between the two investment groups, doing deal flow and learning all about the different companies. So the next 10 years was spent every month with two investment groups, reviewing anywhere from 20 to 40 companies for 10 years. And what I used to do was I used to go to the people in the audience, the investors, after the presentation, and I would say, why did you ask that question? Why was that question so important to you? And then they would go into finance, sales, manufacturing. So I had an education with 50 investors 2 or 3 times a week or 2 or 3 times a month for 10 years. And I really was able to learn quite a bit. And when I went to Boston, there was about 8 investment groups. And Boston's an interesting city. I was from California, so I knew absolutely nothing. I was not from Boston, so there wasn't possibly anything that I could possibly know. So I went to the 8 angel investment groups, and I finally went to Sky Ventures with Yale Giffen. And he said, what do you think of this company? So I told him, and he said, sent me another company. He said, could you write up a little review on it? So what turned into 2 short evaluations turned into every month I was reviewing about 5 to 10 companies for him. He had unbelievable deal flow. He was from Israel, so he was getting stuff from Israel, Europe, and all over the United States, and especially in Boston. So we had it well connected. We worked out our meetings were at Northeastern University where I was, and we had a buffet lunch, and we had the people present at the presidential room at Northeastern. So it was very special. And we got all the people from Harvard and MIT to come, and it turned out to be very good. So it was a very effective and meaningful experience I had in Boston, which I can go into in a lot more details. So let me tell you about some of the scars. One of the students, while I was doing research, worked with me and published several papers with me. He wound up going to medical school and into dermatology. And he approached me about 10 years ago and said that he had identified a set of genes in patients with melanoma that would discriminate moles from melanoma. And there were 7 genes. And we used the RNA PCR assay to discriminate if the moles were expressing those genes or not. And that's when I started working with Manfred Dietl in Germany after being in Germany. So we looked at several companies that we wanted to sell the company to, but they had different issues, and they wanted monoclonal antibodies, and we had RNA PCR, and then we did monoclonal antibodies, and they wanted RNA PCR. Anyway, we published the paper in PNAS, and Myriad Genetics came, and after about 18 months of the negotiations, they set out milestones and royalty payments, and we sold the technology to Myriad Genetics. They now have a kit on the market. It now has 20 genes, and it's part of their portfolio of screening cancer at Myriad Genetics. So my grandchildren will be very happy with the milestones. It's a royalty payment. So the more assays that they do, eventually I will get some money. So when I got to Boston, I realized that one of the problems I had was, as chairman of the Pasadena Angels, was entrepreneurs did not understand the process of funding, and the investors didn't understand the scientists or didn't understand the vocabulary. So I taught a course that about 600 students went through. We did in-house university companies, We evaluated them. The student acts as consultants for those university companies. And then the students submitted at the end of the semester a plan, business plan, an investor presentation, term sheets, and due diligence to 100 CEOs. Now, the majority of the CEOs were very happy with it. But there were a few CEOs that could not believe that those students knew more than they did about their company or their projections. And some of them just threw the whole report into the garbage bin. Yeah. So they're very opinionated CEOs sometimes. So let me tell you about the entrepreneurial ecosystem. Boston has a unique ecosystem. Within a 2-mile radius, you have 6 major universities, and they're all connected by light rail within 20 minutes of each other when they're working and when it's not snowing. So you have Harvard, MIT, Mass General, Tufts, Boston University, and Brigham's, all doing really first-class science and with very strong NIH funding. One of the big jokes when I was on the Sky Ventures, we would screen a bunch of companies, and then we'd have 2 or 3 that we would do 30-minute video conference with. Multiple times, the science was really strong, but it was more like an NIH grant. And so I would ask about the science, and then I would ask about the business. And they got really annoyed because I could ask both the science and the business questions, and I wasn't supposed to do that. So it was an interesting experience, but the science was just unbelievable. In addition, they had all the major pharmaceutical companies in Cambridge or in Kendall Square with venture funds. And their venture funds have no strings attached. They basically give out grants with no strings, no right of first refusal, which was just unbelievable from when I was in the pharmaceutical industry. The venture capital community is very strong there. And as I said, the angel community is very strong. So they have no problems looking at therapeutics. And the system is that the venture capitalists go to the universities, go to the tech transfer office, get the technology, put a team around it, de-risk the product, and then sell it to the pharmaceutical industry. That's the method that has worked for them for the last 10 to 15 years. And in that process, there's a lot of small group of people working on those processes to facilitate the technology coming out of the universities to a business component to be sold to the pharmaceutical industry. The pharmaceutical industry liked it because it was de-risked by this venture capital team, making sure that it worked at least a little bit. They also had multiple incubators and accelerators, both private and public. And then they had a system called Mass Challenge, which was a pretty unbelievable organization. They screened about 1,500 companies nationally and internationally every year with mentors, with screeners, and with an incubator. So it took them about 10 to 15 years to get it up and working properly. But that was the Boston entrepreneurial ecosystem, and I can go into that in more detail. So the technology ecosystem, Boston is clearly a very good place to do biotechnology. San Diego and San Francisco each have their technology sectors. Los Angeles is developing Silicon Beach. We're talking to San Gabriel Valley right now. New York City, Texas, Florida, all these states and cities are putting huge amounts of monies into incubators and venture funds to help stimulate their economy. Austin, Texas is becoming one of the hot areas for biotech. So at a federal level, at a state level, and at a city level, they understand the importance of accelerators and incubators, and they understand the importance of creating that culture of spinning out technologies and companies. And they're all putting money into it over the last 5 to 10 years. So what's the future? Whoop, what did I do?
You click down. Gotta go backwards one slide.
Got it. Okay. Whoop. Okay, so what's the future? Academics is still run by grants. You've got to submit your grants, you got to get your grants, you got to get peer reviewed. That's always going to be a mainstay for very early stage research and basic research. Biotechnology, the investors are willing to take a risk and fund ideas that may come in a product that may become very valuable. And the pharmaceutical industry just needs to keep pumping out their products, their drugs, so their shareholders can get a good return on investment. The venture funds are the same way. They're all looking at what kind of return on investment they're going to get. So where have we been and where are we going? Genentech is the classic story. In the 1980s, biotechnology, they developed proteins instead of small molecules, which disrupted the pharmaceutical industry. The human genome was sequenced in 2000, but we haven't been able to define human disease by DNA. Now that's starting to change a little bit after 20 years. They're starting to see differences in populations and differences in gene expression and differences in the DNA sequencing of different subgroups of the human DNA. The hottest area right now is called CRISPR applications for modifying or changing genes in the cell, but they're still stuck with the same delivery and toxicity problems that has plagued gene therapy for about 40 years. How do you selectively get your therapeutic or CRISPR gene to the right place, to the right cell, and not cause any toxicity? And that's still being studied. So fundamentally, what we don't understand is the molecular pathology of human disease. Is that going to involve RNA processing? It's suggesting that it might. And we need to learn more about how DNA goes to RNA, goes to the protein, goes to the disease. And RNA therapies. You saw from COVID, Moderna, Pfizer, many biotech and investors are going after new RNA therapies for even the common cold or for cancer or for inflammation. And Moderna now is saying that they're hoping to get an RNA cancer vaccine by, well, soon. I think that's a mistake. I think it's 2030, not 2023. So I think it's going to take a while for them, but they're working on it a lot. So that's basically just an overview of my experience and the direction that the field is probably going to be going in. So I'll be glad to take any questions on any of the topics.
You want to unshare your screen so we can see everybody? Put it on gallery view, I guess. I can figure out how to do that. So anybody have any questions? I have questions, but some of them are kind of technical. Yeah, I guess related to entrepreneurship, couple of experiences as an investor that I have, and I don't have that many. The PhD, their lab came up with some great ideas, and then the PhD student graduates and then becomes maybe CEO or CTO of the company that spins out. Your example from Boston was the team went to the tech transfer office, grabbed some technology, and then took it. And to me, that my model was kind of like, well, if you get a PhD student who has some business savvy, they can take it out of the lab and bring it to market. So which is more common?
Okay, my first investor meeting in Boston was Boston Harbor Angels, and I sat next to the chairman, and he said we went through about 4 or 5 companies, and every company had someone affiliated with MIT. I mean, everyone, faculty, postdoc, graduate student. So I turned to him and I said, do all your companies have some affiliation with MIT? He said, yeah, they're the only ones we fund. So their philosophy was, if the science is good enough, which it usually is at Harvard or MIT, they can just put a business person with them and drive the company. So that was their philosophy.
But do they still use the kind of the knowledge of that lab or that PhD student? Because I think I actually with Cal Poly, I was part of their tech transfer office while they briefly had it. And it was like they hand us a bunch of patents and say, are any of these worth anything? And we're like going, I don't know. You
Well,
can't look at a patent title in an abstract and not be in that field and know whether it has any value.
what you see, what they have is they have seasoned CEOs, they have seasoned COOs in the venture community. They have a group of people that they can draw on. And so the scientist comes in, and the scientist's job is to do the science, and the CEO's job is to de-risk the technology and sell it to the pharmaceutical company.
Okay. Which means, I guess what I was saying is the scientist is coming out of the lab and knows the technology really well, knows the science really well, and then they get a team that helps them commercialize it.
Yeah, it's in my experience, there have been very few scientists that have been really successful in being able to go to the different stages. So in my book, I say, are you an inventor? Are you an entrepreneur? Or are you a CEO? And each one has different skill sets. And it's hard to transition from inventor to entrepreneur to CEO. It's done, and you have to have good mentors and you have to have a good advisory board. But that skill set is really unique.
Kevin, I have a question. This may not be quite in your wheelhouse, but what do you see as the obstacles to getting cancer, um, ways to prevent cancer or to treat cancer? What are the obstacles that we're running into that we spent so many years, so many millions of dollars? What seems to be the problem or the obstacles there?
There's a lot. The easiest way to reduce cancer is stop smoking, environmental factors, early screening. So if we do those, that would cut down at least half the cancers. The second problem is we've just approached the problem the wrong way. And I've been in the field for a long time. We had the wrong models. Initially, the models were mice with leukemia. And the chemotherapy agents that worked with children, pediatric cancers are really fast growing. And mice had leukemia, are really fast growing tumors. So when you gave a toxic drug, it killed the fast growing cells. So we didn't get any selectivity. The big problem with gene therapy, everybody wanted to do gene therapy for the last 20 or 30 years. There was a delivery issue and there was a toxicity issue. So the question is, can you identify the real driver of what's causing the normal cell to become a cancer cell? Can you differentiate that normal cell from cancer cell? And can you knock out the gene in the cancer cell and not harm the normal cell? And that was a big problem. So I'll give you an example. There's a term called ribozyme. It cuts RNA. It's very similar to CRISPR. And one of the major cancer oncogenes that causes cancer is called the RAS oncogene. And the RAS oncogene is mutated at codon 13 from GAC to GUC. And ribozymes cut GUC. So I was a smart ass. I put it into a virus and I put it into a mouse that had a RAS mutation with cancer in a bladder cancer, and the cancer went back to normal. All the clinicians went berserk. I repeated the experiment several times. I've published about 10 papers on it. You can knock out the driving oncogene with an antisense ribozyme or CRISPR, and it will reverse the process of the cancer cells. However, you cannot deliver it in a high enough dose in a patient to be effective because it gets inactivated. That's been the problem.
Because it becomes what?
It's it's a pharmacological issue. You cannot, well, you take an aspirin, aspirin goes through your whole body and it works. If you have gene therapy, it gets cleared in your liver or your bladder before you can do anything. You can only do what's called local regional treatment of the disease. And at that point, it really doesn't make any difference because in metastasis, you're not going to be able to treat it. So we still don't understand the process of transformation of the normal cell into a cancer cell. We don't know the driving genes that are doing it, and we don't know the molecular basis for doing it. And that's been the big problem. And then pharmacologically, we don't have the right drug. We have drugs that'll cure cancer, but it'll kill the patient. That's what the problem has always been.
So isn't much of it what's going on immunotherapy, where you're trying to cause the body to then suddenly recognize the cancer cell as a foreign?
Yes, immuno-oncology. I know the guy, Ari, over at UCLA. I've been following his work for 25 years. He was one of the people on our cancer gene therapy journal. Nobody believed he could get it to work. It was really just a crapshoot that he finally worked on it. And guess what happened? He got it to work. And it works really well. That's the good news. The bad news is it only works in some types of tumors, and it only works sometimes in 50% of those tumors. This is what happened 70 years ago in Boston. Children had leukemia. They gave them a vitamin. The vitamin made the leukemia grow faster. So Dana-Farber asked, can you make an antivitamin? So Lilly made an antivitamin. They gave the kids the antivitamin. Leukemia was cured in the kids. 44 kids, 1948. What happened? Well, 22 were cured. 22 didn't respond. And within 18 to 36 months, the 22 that responded relapsed and had the leukemia again. So the same with immuno-oncology. They're not exactly sure how it works, but it works. And they're very excited on why 50% of this component of this type of cancer works. But the other question they have to ask is, why is it not working in the other 50%? So it's a really, really big step. It's a significant impact on treating patients with cancer, but it's not a cure.
And the DNA cancer vaccine or the RNA cancer vaccine, is that an immunotherapy type approach, or is that?
Yeah, well, what they're saying is, look, we got it to work for a virus. The vaccine, the COVID vaccine, is basically an RNA virus, which I have to tell you is just remarkable, the story, how they got that to work in such a short time. Now what they're saying is, well, if we can do that with COVID and that virus, why can't we do it with other viruses? Or why can't we do it with other diseases? But it gets back to Jim's question. How do you know which is the right target to knock out and bind to? So it's, again, a selective, can we selectively find the tumor gene that's different from the normal gene?
Any other questions? Chris, you're presenting tomorrow. You must have questions.
Lots of questions. What are you thinking is the delivery system going to be? Because that we haven't figured out, right? We're still playing with the same viruses and the same liposomes and whatnot.
Yes, I have
Nothing has
done all the
changed.
viruses. I have done all the viruses delivering ribozymes to treat the patient. I have done some of the lipid delivery systems. Now, they are getting better. The nanomolecules with the lipid delivery systems are getting much better. But again, Why would the cell want to suck that up? It comes down to a fundamental question. How can you make the cell take up the nanoparticle that you want to deliver it to the right part of the gene to knock out that gene? That's called selectivity. And that's what the problem is. It's very hard to understand it. How to get it into the cell, how to get it to the right place in the cell, how to shut down the right gene.
Well, I'm actually an endocytosis guy from my background, scientific background, and the issue is that everything goes in. Actually, getting into the cell is very easy, right? You feed the cell nanoparticles, it takes up nanoparticles, but getting to the right place.
Yep, and it can also spit it out, so it can
And spit it out and degrade it, and
yeah, right.
yeah.
So yeah, it's not stupid. So
definitely not.
so one of the things that we did was we worked with a drug called cisplatinum and a nucleotide inhibitor. And with cisplatinum and AZT.
Mm-hmm.
And we found out that cells resistant to cisplatinum spit out a lot of DNA. So when you gave them platinum, the cell just spontaneously started spitting out DNA to bind the cisplatinum so it wouldn't harm the cell. So when we gave cisplatinum and AZT, it chain terminated the DNA repair and it killed the cancer cell.
Wow.
So you tricked it. Yes. And that was the treatment that they used on the last emperor in Japan when he had cancer, because it was very low toxicity, but it was effective.
What did he end up dying of? Not cancer, I guess.
Well, he eventually wound up dying of cancer because they didn't want, they have a different philosophy of treating patients with cancer in Japan. But what they wanted to do was to show that, they were trying to do something and they were trying to do it without toxicity. So that protocol minimized the toxicity, but you had to give a higher dose within 18 to 26 months, and they didn't want to do it.
Anybody else have any?
I guess I have a question about, oh, sorry, Mark, you're on mute. You go.
Oh, I muted myself. All right, there we go. Two-parter. Why do you keep coming back to Pasadena? And actually, more importantly, now that you are in Pasadena, you mentioned the San Gabriel Valley. Where do you see the kinds of things that you're doing, be they research, manufacturing, whatever, evolving in the San Gabriel Valley economic system?
Well, okay, so I really can't answer you why. I always call it divine intervention. That I keep coming back to Pasadena. I don't have a choice in this. It's just divine intervention. Of course, my wife helps a little bit on that.
Okay. But now that you're here, how is what you're doing going to involve the different cities in the San Gabriel Valley?
Well, I am a member again of the Pasadena Angels, and I am working with Bruce, and we are having this conference here today. So why is Boston so successful? Because people are educated to driving Boston as one of the top places for biotechnology. That's what you have to do here in San Gabriel. It doesn't have to be biotechnology, but it's got to be some innovative technology that's going to be useful for the next 10 to 50 years. So I'm only trying to help the entrepreneurs. I saw the failure rate of entrepreneurs 20 years ago was appalling. So I said to myself, how can I help the entrepreneur be more successful? How can I help the investor understand the entrepreneur better? So that's why I wrote the book.
Yeah, we don't quite have 6 colleges, major universities within 2 miles of each other, but we do have Harvey Mudd on one end and Caltech on the other end, and then there's USC and UCLA. So and then there's City of Hope, which actually has a graduate program, and I don't know if the other Claremont colleges come in or not. What's that?
I said you got Cal Poly out here too. So you have a number of universities. They're not 2 miles away, but they're freeway close.
And you have
Yeah.
Citrus with their biotech programs that they have.
Yeah, so you need a thought leader that will bring some continuity. That's what they had in Boston. They had a respect for all. There was a lot of competition among them.
Yeah.
But there was a lot of respect that, what is best for Boston and then what is best for Harvard or MIT and the investors say, how are we going to make money? Well, we're going to make money because we're going to take really good science out of the universities and commercialize it. So what we need is really good science and commercialize it. And why does it work? Just takes one. Professor Lerner at MIT has spun out 200 companies from associated, not all from MIT. One of them was Moderna.
Hmm.
So what does that say when an investor comes to Professor Lerner's office? Whatever you're doing, I'll pay for it. So that's kind of the culture that you need in San Gabriel Valley. You need a kind of a track record of spinning out technologies that are successful.
I think we have some of that, but it's not at that scale. Go ahead, John.
I come from the electronics industry, and over the years, I've seen entrepreneurs, and most of them have in their mind that I can make a big company. And the models are Hewlett Packard, Apple Computer. In the software era, there was even less capital required to get started. So there was Microsoft and Facebook. And everybody that I see in biotech, maybe Chris Booster can make a company that he can stay with for a long time because he's going to sell microscopes to people. But most of the stuff you're talking about, Kevin, a small company, even with a bunch of investment, can't get it to the market. It seems like the goal over and over again is to sell to a big company. And if you're interested in regional development, those big companies that are going to buy you maybe are way across the country. So, I mean, is there any opportunities for new biotechs to make a lasting company instead of just one that gets sold to big pharma?
Well, you are right. The difference between biotechnology and software, even for investors, is a big choice. You can get a bigger home run with a therapeutic, and that's what they do in Boston. They have no fear of FDA and doing a therapeutic. I did a diagnostic company, and they looked at me and they said the return on investment is not high enough for us to be interested. So here you've got fear of FDA, therapeutics, diagnostics. It's going to take a long time to develop. Software, it's an app, it's easy to apply, it's cheap to develop and grow. Now, how can you get a biotech startup company here to grow? Co-development deals. So, well, there's three choices when you look at the pharmaceutical industry. One, you get bought out. Two, you can co-develop. Or three, they can make an investment and just follow you. So the best way to do it is co-development. And they prefer not to risk a lot of money. So co-development is a cheap way to do it for them. They put a little money in, they get to watch what you're doing, they can reproduce it in their own lab, or they can help you develop it in-house here in the San Gabriel Valley. So I would go with co-development as an option.
Kevin, if I may, to your point, I had a client a few years ago out of Caltech who developed, I forget exactly what it was, but it was a biomedical product or whatever, got some funding from one of the major pharmacy companies. And then a couple of years later, they bought him out. So he wasn't able to develop the company, but he developed this product and then he, moved into that company and he's been working with them. So that's what happened right here in the San Gabriel Valley.
Okay, so I can tell you from the other side of the table, when I sat in the pharmaceutical industry, we would never do that. And the reason we would never do that was it's cheaper to have him develop it. It's cheaper for him to develop it because if I was to take all my resources of my department and buy them out and then reproduce the whole thing in my lab or my department, it would be insanely expensive. So what I did, and one of the things that really they always gave me goals, and then they gave me stretch goals. And I was always able to get to my stretch goals because I licensed it out or funded out people in academics to do the work, to spin out the products and develop the products, and they would get more money the faster they did it. So from a financial point of view, it's cheaper for the pharmaceutical industry to co-develop and let them take the risk of even growing the company. The question is, from the pharmaceutical point of view, does your company have the maturity to grow? As I said, inventor, entrepreneur, CEO. So that's where they get a little skittish when they look at co-developing, and giving the founder enough support so that he can grow the company big. But it does happen. It's not a lot, but it does happen.
One observation, somebody was like in one of the top accounting firms who came to one of our meetings. I thought he had a great observation. He says, in Los Angeles, we don't build big companies, we start them. So if you have a successful exit, hopefully the entrepreneur founder stays in the area and starts another company. That's what's going on in Silicon Valley. They just keep starting new companies because the founders get seeded into the ecosystem.
It's a track record. They have a track record.
Yeah.
So what we found out in the Pasadena, so let's go back to that a minute. So you get an inventor, then you get an entrepreneur. The entrepreneur is successful. He sells his company. In general, most investors will fund his second venture. And most times that venture fails. And the reason it fails, because we've done some studies on it, is the skill set for the entrepreneur to be successful in the first company was a set of things that he did, and he did right. The second company, there was no guarantee that he was going to be doing the same problem with the same issues, and he couldn't pivot enough. And we find out more than likely, the second company fails.
Right. So I have a really, patent, a question only a patent attorney would appreciate. As you said, your melanoma company had 7 genes. I forget the name of the case that took away the ability to patent genes themselves.
Yes.
So would that same venture have worked under the new law? And how did Myrid, how do they capture 20 genes and have a patentable assay? How do they get around? And just for background, you can't patent natural things. And that's always been wrong. But it became quite clear in a Supreme Court case several years ago that affected gene therapies.
So when I was in the pharmaceutical industry, we had the same dilemma. Everybody was sequencing DNA. So there were 2 positions in the pharmaceutical company. The first position was taken by a DNA sequencer and said, I want to sequence all the DNA. I, on the other hand, said, no, we're going to use ribozymes and inactivate genes to see if that plays a role in the cancer, because then you would have function. The argument against that was it was going to take a long time. Conversely, if you just sequenced all the DNA, you could claim everything. Goes to your point. So the Germans agonized over this for 6 months. And at the end of 6 months, they brought us both in. And you know what their choice was? They funded us both for another 6 months.
Right.
At the end of the 6 months, what did they do? They called us both in again, and they did it again. So for a year and a half, we kept collecting data. And finally, what they agreed to was they wanted the function, not DNA, because of your reasoning. You can't you can't patent DNA. And that's what we did with melanoma diagnostics. We showed that if you knocked out those genes, then they wouldn't be cancer. So they played a role in cancer, and they were upregulated in the cancer cell and not upregulated in the normal cell. So we were able to show function of the gene played a role in the disease of the melanoma.
So you had a process, basically,
Yeah.
process patent.
Yep.
Any other questions, Chris? You have a presentation tomorrow. You need to get all the questions answered.
Yeah, those are different types of questions for tomorrow. No, but I have actually one on innovation ecosystems. Particularly what I'm noticing, the way you put it, the initial innovation comes out of academia, but me as an academic or an academic who's running for the exit, what I see in academia is that when we started measuring everything, and now that we're measuring everything, the drive is to reduce risk in academia as well, and only fund the things that work. So we're only funding the obvious stuff that, right? We can see around one turn, and that's what we're going to fund. We're never going to fund the things that require us to see around 2, 3, 4, 5 turns. And it's getting worse and worse, it seems. Do you see that the same way?
Okay, let me tell you about RNA PCR assay. I did the RNA PCR assay with no Taq polymerase, with no PCR machine, in 2 water baths with 8 scientists. That manuscript was rejected from 10 journals. Over a 3-year period, I was told it was impossible to do. Finally, the editor of Cancer Research called me up and said, Kevin, we're publishing your paper. Just answer the reviewer's questions. It took 3 years.
Yeah.
I had my NIH grant rejected for the RNA PCR assay. So it has always been like that. They're very conservative. That's why I went into biotechnology, because they're more willing to take a risk. But academics does fundamental research.
Mm-hmm.
And the fundamental research doesn't necessarily lead to a product, but the fundamental research can lead to insights that could possibly make a valuable product. So that's the difference. And biotechnology is willing to take the risk. Startup companies are willing to take the risk.
Yes, indeed. Exactly why I'm doing what I'm doing. Yeah, but it's frustrating because if you can see around 2 or 3 corners, I just got an SBIR grant slapped around my head. I didn't get it funded exactly because they don't see that far, and they're not willing to take any risks, even though it's called innovation research, right?
I can give you advice on that. Just answer the reviewer's questions and send it back in.
That's exactly what I'm going to do.
So that's the good news. The bad news is they only sit on that review for 2 or 3 years. So the guy that turned down your grant may be off, and now you have another guy that'll do the same thing. So then you just have to. When I was in academics, I was submitting 10 grants a year. I was getting 1 or 2 funded.
Yep.
When I went into the pharmaceutical industry, I told my boss, I said, I'm having grant writing withdrawal. Because the first year I wrote no grants. I couldn't believe it. It was just, I was driven insane. So yeah, so that's part of the problem. That's part of the process.
Kim, do you have a question? You just unmuted yourself.
Yeah, if it's yeah, thanks, Bruce. Hi, Kevin. Is it okay to ask an angel investor question?
Oh, sure.
Yeah.
So you told me a while ago, well, years ago, when I last saw you with the Pasadena Angels, that medical devices are not a good prospect for angel investors to invest in, but I don't know why.
Okay. Yeah.
And then I do have a second question. Your last slide on what is the future, you had different types of life science technologies listed there. Which ones of them, I think you had investors behind only one of them, biotechnology. Are the others, could they be possible candidates for angel investors to participate in as well? So that's my second question.
Okay, so in the Pasadena Angels, there are about 100 investors. The majority of them are not biotechnology experts. So the investor is going to put his money in to get a good return on investment as quickly as possible. And as we discussed earlier, a software application doesn't cost a lot of money and potentially could get you a good return, 2 to 5-fold, 10-fold return on investment. A biotechnology, a diagnostic would take, well, let's take melanoma diagnostics, for example. We spun the technology out of UCSF and we wanted to flip the company in 2 years. All right. We wanted a return on investment in 2 years. We wanted to sell it. Problem one, we ran into problems getting the technology out of UCSF. Problem two, we ran into problems when we outsourced making the monoclonal antibody. Problem number three, most of the people did not want to invest in a company that had not published their data. So we published the data, we got people to come in, and then when we negotiated the deal with Myriad Genetics, the return on investment was out over a 10-year period. It was milestones, royalty payments. So if the investors put money in, it was going to be over a 10-year period that they would get the money back. Therapeutics in Boston is much more popular because the amount of money that you need to put in and the amount of money you would get from a drug that you would sell over the counter is enormous. So you get an enormous return on investment. So that's why in Boston, they don't like to invest in diagnostic companies. In California, they do like to invest in diagnostic companies, but they're prepared for a long wait for their return on investment. And most angel investors don't want to wait that long. We have a certain amount of money, and we need to, if we're going to reinvest, we need to get that return on investment as soon as possible.
But we have, Pasadena Angels has invested a lot in a lot of healthcare, biotech type companies. So it's kind of like, we don't do that. We just did it again. Probably 3 companies a year, I would think.
Yeah. So, I mean, yeah, there's a big difference. Whereas in Boston, every company, 9 out of 10 companies I saw were biotechnology companies. So that there's a more broader portfolio here in Los Angeles. And each individual investor has risk averse investments. So when you say biotechnology, there's a red flag comes up here in California, but there are other reasons they may or may not invest in a biotech company. It's really an individual basis.
And there was somebody that presented, I forget, I'm blanking on her name. You probably know her. She's a TCA member out of Riverside, or a biotechnology person. And she basically said, you're not looking to go public. You're looking to get into clinical trials. And then once you get to clinical trials and have some data, then you sell. So the early investors their time horizon is get that first positive result at clinical trials, which doesn't make it not risky. It just means that you're not really contemplating the long haul. You're not contemplating phase 3 trials. You're contemplating phase 1 trials. Is that kind of true, Devin?
Yeah. Well, I'll give you another example in Boston. One of the faculty members I was with in Boston told me his story, which was a really fascinating story. He was an academic. He was doing research on embryo development, and he started an in vitro clinic. And I said, that was a little risky, wasn't it? He says, well, he got some mentors, and he went around and he talked to a lot of people, and he got himself a really nice group of advisors, and he had a really good team, and they commercialized it, and they set up clinics. They had 2 clinics where they could do in vitro fertilization based on his research. And they had a CEO. Everything was going smoothly. 10 years in, they were ready to start growing and getting more clinics. And the technology did not work when you scaled it up. And he lost everything. So those are the risks. But he did a great job over that 10-year period. And it was, sometimes it's just out of your control. What can happen?
So one more question on I've heard academics, including one that's a young academic that happens to be related to me, talking about going from academia to pharmacies to big pharma. And there's just this sellout, this notion that if you go to big pharma from academia, you're like, it's a horrible thing to do. So what was your experience in going from the lab to big pharma? It sounds like it was a very positive experience and you found it, I guess, liberating, or was the bureaucracy, or how did it change? And is it good or bad, or what, something else?
Okay. So first, it was my mentor, the CEO of the company that I knew for 10 years. So that helped. And he didn't want me to fail. The second issue was, the best way to put it is the pharmaceutical company has to make money, and it has a portfolio, and that portfolio has to sell its product and have patients use it and be successful. If the research that you're doing aligns with that portfolio and that pipeline, it's perfect. There's more than enough money to do whatever you want to do. The problem is it changes every 6 to 12 months. Every 6 months, I went to Berlin with 40 people in a 30,000 person company, and we set the goals of what the next 6 months were going to be. And you had to hit those goals. And it turns out that bonuses are very important with those goals. So the goals had to be met to get the bonuses to make everybody happy. And it had to be aligned with the portfolio and the pipeline of the pharmaceutical company. And that's always changed. We had a joke in the pharmaceutical company with the chief medical officer. He was known as Dr. No. He would say no to every proposal that would come through. And you know what? He was right 9 times out of 10. But after about 5 years, he would leave and move to another company. And guess what he would do for the next 5 years? Say no. And he was very successful.
Right. So yeah, I guess you can't pursue things because they need to be pursued. You pursue them because the corporation says this is what we're going to be doing.
Ultimately, you have to address the shareholders. The shareholders need their investment. The pharmaceutical company has to sell the drugs. Pharmaceutical company has to make money to pay for everything. Now, my budget was just astronomical. My travel and entertainment budget was better than any salary I've ever had anywhere before in my entire life. I mean, I would have cars pick me up. I would fly business first class everywhere I went. I mean, it was just amazing. And it was all making sure that my job was to make sure that the science validated the concepts that could go into the pipeline, which would then become candidates that would then go to the product pipeline, which they could sell. Now, I'll give you another example. One day I walked into the department, and there was this German woman sitting there, and she said, "I've been transferred from Berlin." I said, "Oh, that's good." She goes, "Yeah, I'm in your department now." Like, "Oh, that's good." So I gave her a lab bench and I let her start working. Within 6 months, she had produced an enormous amount of data. So I gave her 2 technicians. 6 months later, she had produced a concept for a diagnostic for cancer using a vitamin. 6 months after that, she had it in a clinical study. And yes, 18 months from sitting in my department to getting the first patient clinical data. This is a woman that nobody she couldn't work with anybody in Berlin. They were all the guys. They didn't want to have any women, to deal with. So they sent her over to me.
Wow.
And with me, she became so successful that they were offering her everything under the sun to come back.
Good.
So those are some of the stories of what the pharmaceutical industry is like. She had the resources to pursue it because I needed a diagnostic in the pipeline to work, and she got it to work.
Right. Anybody have any last questions? We're at 8 o'clock.
I just have one. I'm wondering if when we were talking about the ecosystem and how Boston has so much leadership in developing technologies, do they also have the pipeline? I'm just wondering, when we look at like the combination of City of Hope, Caltech, Citrus College, Cal Poly Pomona, if that like that you're filling all the roles makes a difference versus just having people that are, the scientists at the top.
Okay, so the good news about Boston is you have all these research laboratories doing all this kind of research. You have these major pharmaceutical companies with their portfolios. So the venture capital sits right in the middle. So the challenge for the venture capital is, what does the pharmaceutical company need for their pipeline? What does the research going on at MIT and Mass General and Harvard going on? And where does that all fit in? And how do I align them to get that pipeline to go through? And I have to tell you, there are some really, really clever people in Boston doing that. Eyal, the guy I worked with Sky Ventures, was able to align that all the time. But that was a full-time job for him for deal flow, dealing with academics, dealing with other angels in venture capital, and then finding out what the pharmaceutical industry wanted. What made Sky Ventures so successful was everyone was treated really well every month on the 17th floor of the building in downtown Boston for the president's conference room with a buffet. So 100 to 150 people showed up every month, networked, talked, and told you everything that you needed to do. And that's why they love coming to Northeastern, and because nobody at MIT or Harvard or Mass General or Brigham's was ever able to do that. We were able to do that because the president and the provost wanted those people in on campus. So it was putting those people together in a cultural environment where they felt safe and they could talk to each other, and they were having a good time and they were exchanging information. That's what made it work so well.
Any other last questions? If not, I thank Kevin for presenting. It was very, very informative. Appreciate it.
Thank you. Glad you came out.
Thank you.
Thank you, Kevin. Great presentation.